Thank you for inviting me to give the talk. I'm going to talk about using electronic health records to test the hypothesis generated using molecular data and then integrating these two sources of data to really accelerate clinical translation. So when you look at the biomedical researchers communities, there are largely speaking two communities. One group that really studies the science of medicine, so the biological underlying biological mechanisms using molecular data like gene expression, proteomics, jivas, and so on. And then the other community that studies practice of medicine using electronic health records. And when you look at these two communities, there is little to no overlap. People who do molecular research, they don't really analyze EHRs, and those who do EHR analysis, they don't usually go and look at the mechanism. So we asked, can we break these silos and integrate both science and practice of medicine so that it would actually accelerate clinical translation? So to sort of test this hypothesis, we started with publicly available gene expression data from blood sample of patients with IPF, idiopathic pulmonary fibrosis. This data was published in Science Translational Medicine a few years ago. So we used this data, applied the deconvolution methods developed in my lab, estimated proportion of different immune cells in each sample, and found that higher proportion of monocytes was associated with adverse outcome in patients with IPF. So now we needed independent cohort to validate this observation that we had made in this publicly available cohort using molecular data. So we decided to look at electronic health records because complete blood count is a routinely carried out test since mid 1950s across virtually every clinic in every country in the world. So we started at with Stanford Electronic Health Records in collaboration with Nigam's group. We looked at all the IPF patients and their absolute monocyte count within the first 30 days of diagnosis, and we chose a threshold of 0.95 kilocells per microliter, just because that's what the medical textbook says is the threshold for abnormally high monocyte count. And when we looked in a Stanford cohort, indeed, absolute monocyte count greater than 0.95 kilocells per microliter at the time of diagnosis predicts is associated with reduced survival. But this is one small cohort, only 130 samples. So we sent our code to Joshua Danny at Vanderbilt, and he sent us a figure showing that in their 1600 patients, the same threshold was still associated with reduced survival in patients with IPF. And then through population health sciences and the Nigam's group, we analyzed Optum, which is the countrywide electronic health record data. And again, same threshold, absolute monocyte count greater than 0.95 kilocells per microliter at the time of diagnosis was associated with reduced survival in patients with IPF. So next we ask, is this only specific to fibrosis in lung, or does this association also holds true across the other fibrotic diseases? So we repeated the same analysis in 3 other fibrotic diseases: hypertrophic cardiomyopathy, systemic sclerosis, and myelofibrosis. So heart fibrosis, skin and other internal organ fibrosis, and bone marrow fibrosis. And again, across all 3 data sources, all 3 diseases, absolute monocyte count greater than 0.95 kilocells per microliter at the time of diagnosis was associated with reduced survival. So really robust result. And overall, what we were able to show is that irrespective of how you measure monocytes or how you diagnose a fibrotic disease, higher monocyte count at the time of diagnosis was associated with reduced survival. But you can say, this is our analysis, we might have done it so many times, can somebody else reproduce it? And less than a year after our paper came out, a group in Australia repeated the same analysis in IPF patients from 3 Australian states, used the same threshold as we had described, and showed that indeed absolute monocyte count, higher absolute monocyte count at the time of diagnosis was associated with poor survival. And then a group in Europe just earlier this year published another paper in the Blue Journal using patients that were enrolled for phase 3 clinical trials for treatment of a new treatment of IPF, that indeed the patients at their higher monocyte at the time of enrollment was associated with poor survival. I'll stop here by really thanking the people who did this work. So Madeline Scott was the one who led the project in collaboration with Katie Quinn from Nigam's group, and all of our other collaborators who really very graciously provided this data for us to analyze and made this a very successful collaboration. Thank you.